So, why exactly does inflammatory bowel disease dramatically increase the risk of colon cancer? It's honestly a macro-level medical mystery that has stumped researchers and doctors for years. I mean, we've known for a long time that there's a definitive link there, but the actual biological bridge connecting these two distinct conditions, well, that's been hiding in the microscopic world of our cells. Just look at these striking contrast here. You have a normal, balanced gut environment that abruptly shifts into this fiery, inflamed, and eventually cancerous state. In today's explainer, we're literally going to uncover the exact biological mechanism, the missing link, if you will, that bridges these two states. We are tracking down the rogue cellular engine that drives this really dangerous transformation. So let's get right into it. Today, we are unraveling a massive breakthrough, the medical mystery of HKDC1, its connection to the microbiome, inflammation, and colon cancer. Get ready because what we're about to explore is going to completely change how we view the ecosystem living right inside our own digestive tracts. Here is our roadmap for the explainer today. First, the colon cancer pipeline. Second, meet the fifth hexokinase. Third, fueling tumor growth. Fourth, the microbiome connection. And finally, targeting the future. Section 1. The colon cancer pipeline. The IBD to cancer link. For individuals suffering from inflammatory bowel disease, or IBD, there is a very well-documented, significantly increased risk of developing colorectal cancer. It's a pipeline the medical community has observed for decades. But honestly, the frustrating part has always been the why. How does chronic inflammation in the intestines directly instruct healthy cells to mutate and form tumors? The precise molecular mechanisms, the actual chemical dominoes falling in this sequence, have remained largely a mystery until some incredibly recent breakthroughs. Section 2. Meet the fifth hexokinase. Introducing HKDC1. Okay, meet the prime suspect in our biological detective story, HKDC1. That stands for hexokinase domain containing 1. To wrap your head around this, think of a hexokinase like a crucial spark plug in a cellular engine. Its normal day job is to process cellular fuel, specifically glucose, to keep our cells running smoothly. It catalyzes the very first step of glucose utilization. For a really long time, we thought there were only 4 of these hexokinase enzymes in mammals, but recent genetic analysis, it revealed this hidden 5th hexokinase. Check out the dark brown staining in these tissue samples. That represents the physical location of the HKDC1 protein. It is heavily localized right in the intestinal brush border epithelium. Basically, it sits exactly at the boundary of our intestines, precisely where your gut meets all the outside materials passing through. It's perfectly positioned to play a major role in gut health and, unfortunately, gut disease. Section 3. Fueling tumor growth. The rogue engine. Let's map out this disaster sequence step by step. Step 1. In states of inflammation or cancer, HKDC1 is abnormally overexpressed. There is just way too much of it. Step 2. Because it processes glucose, having too much of it acts like a fuel pump stuck on max, driving hyperactive glycolysis. It's just flooding the area with energy. Step 3. All this metabolic chaos activates the WNT/beta-catenin signaling pathway, which is a really notorious chain of events in cancer biology. And finally, step 4, this chemically forces the cancer cells to rapidly proliferate, migrate, and even evade the body's immune system. It's basically the perfect storm for tumor growth. And these mouse models provide some undeniable physical proof of this. Look at the group where researchers artificially decreased HKDC1 expression, labeled here as shy HKDC1. Those tumors are tiny compared to the control. But when they artificially increased or overexpressed HKDC1, labeled OV HKDC1, wow, the tumors grew massively larger. It is an absolutely undeniable direct correlation. So the massive, hard-hitting takeaway here is simply this: remove the protein, stop the tumor. If this enzyme is missing, tumors either cannot develop at all or their growth is drastically slowed down. Grasping this absolute necessity of HKDC1 for tumor survival is going to be the real key to unlocking new treatments. Which brings us to the twist. Section 4, the microbiome connection, the major accomplice. This research highlights a massive revelation. Let me just read this quote. The bacteria in the gut produced enzymes, which can switch off active substances. Hexokinase family members can be regulated by the microbiome. Let that sink in. This means the actual switches controlling this cancer-causing protein aren't just embedded within our own human DNA. They're being actively operated by the trillions of bacteria living inside our digestive tract. To really understand how this works, we have to look at the chemical dialogue happening inside the gut. First off, just the mere presence of gut microbiota directly stimulates hexokinase expression. We know this for sure because when researchers looked at germ-free mice bred to have absolutely zero gut bacteria, their hexokinase enzyme levels were drastically altered compared to normal mice. Furthermore, microbial metabolites, these are the chemical byproducts that bacteria produce, things like butyrate, actually act as master regulators. They can naturally downregulate and suppress these dangerous proteins. Basically, our bacteria are actively dictating whether our cancer engines run hot or cold. Section 5. Targeting the future. Treatment avenues. All of this data opens up two totally revolutionary treatment avenues. On one side, we have chemical inhibitors. Researchers are testing targeted drugs like a compound called NSC-24500 to artificially block HKDC1 and basically starve the colon cancer cells of their hyperactive fuel supply. On the other hand, we have microbiome therapies. Imagine deploying specific designer gut bacteria or metabolites to naturally suppress the enzyme's expression. By doing this, there's an incredible potential to treat the chronic inflammation of IBD and prevent the onset of colon cancer simultaneously just by altering the gut flora. It's a real game changer. Which leaves us with this final, really profound question. If our gut bacteria control the switches for cancer, could the ultimate cancer therapy be hiding right now inside our own microbiome? It's a wild, fascinating thought that totally blurs the line between human biology and the microbial world we carry around with us every single day. Thank you so much for joining me on this explainer. I really hope this structured look at the data has empowered you with a totally new understanding of your own internal ecosystem. Keep learning, keep questioning, and I'll catch you on the next one.